I'm delighted to introduce really one of my favorite colleagues in my department, which is the Department of Biochemistry, Dr. Tim Griffin. And Tim joined the faculty about 15 years ago after doing a very successful postdoc with one of the fathers of quantitative proteomics, Rudy Aebersold at the University of Washington. And since 2012, Tim has been the faculty director of the Center of Mass Spectrometry and Proteomics, and he really turned what had already been a very highly functioning core facility into a national powerhouse. And I don't think I'm exaggerating, but we have some of the best quantitative proteomics that's done in the country. We have people from Vanderbilt coming here and UPenn. And this really has all been under the leadership of Tim. So Tim's newest project is to integrate big data sets, RNA-seq data and proteomic data sets. And so that's known as proteogenomics, and he works on trying to develop user-friendly tools to make this accessible to all of us. And this is supported by a large U24 grant from the NCI. And many of you might be aware that we just put in the renewal of our Cancer Center Support Grant, and one of the newest developing resources is a proteogenomics resource. So we really hope that we can make what Tim is working on available to all Cancer Center members at a subsidized price. So without further ado, thank you for being here, Tim.
All right, thank you, Anya. Very nice introduction. I think you deserve a part in that whole quantitative proteomics because people like you bring good projects to the core facility that actually make us think about new things and new ways to do things. So thank you. Thank you for being here today. I was trying to frantically shovel my driveway before I got here, so at least I think I'm warmed up and ready to go that way, which is a good thing. I'm going to be talking. I'm from the biochemistry department, is where my home is, and talking about some of the work that we've been doing in this area of proteogenomics. So what this talk will be is mostly sort of a description of a methodological approach, heavily in sort of bioinformatics, and really my goal here is to try to get people seeing what this work is that we're doing, but I hope also to see sort of where this maybe could go and what the applications are. So part of the reason this is methodological is that we have been really trying to develop these tools and still are, and now we're just getting to a point where we're trying to apply this, and I hope with coming presentations we're going to have a little bit more on the side of some sort of real data to show that is outcomes of using some of these things we're developing. But for today, it's going to be sort of trying to define this idea of what this concept of proteogenomics is, and something about this platform called Galaxy P, and a little bit of a tour through these tools and what this one of these workflows is, and give you a sense of what it potentially can do, and then a few thoughts for the future. So very happy to be here today, and thank you to Anya for inviting me, because it's a good audience to sort of present some of this work to. Just a few acknowledgments at the start. So the work you're going to see is really a good 5-6 years or more of sort of time and effort, and it's been very much a team effort. I'll hit on a few of the folks from my lab that have really been sort of working on pieces of this as I go through it, as well as Pratik, who's here in the audience, who's really helped kind of co-lead this Galaxy P project since its inception about 6 years ago or so. And also I'll touch on a few other things, but a few others in the audience who are here. We work heavily with the Supercomputing Institute here, who help us with some of the development of software, and JJ, James Johnson, or JJ as most know him, is here. And we also have been doing this work in sort of collaboration, and it's been really actually rewarding that way, that I'll get into this a little bit, but there's this very thriving community out there in sort of this bioinformatics area of people developing software tools, but really doing this in a team and collaborative way. So there's not a lot of fighting as to, I'm going to create my great stuff and nobody else can use it or get credit for it, but lots of interaction and collaboration, which has been terrific. And then the funding, we were fortunate to get funded at the start of this by the NSF, and now more recently through the NCI, so that's helping kind of drive a lot of this. So what I'm going to do is I want to just kind of set the stage and the context here and talk you through, just so everybody's on the same page, sort of proteomics. Mass spectrometry is really the core of this, and that's really going to be the center of everything that you see that we're developing. So I want to kind of just go through this relatively quickly, but make sure that people are up to speed on how we actually do this, because this will put into context sort of the work we're doing. And definitely as we go, if you have some questions that pop up, feel free to ask them. So many of you may be familiar with this, but real quickly, this is sort of the methodology we use in what we call shotgun proteomics. And it's really the sort of driving way we do protein analysis on complex mixtures using a mass spectrometer. So we take mixtures of proteins, we break them into smaller peptides. We use trypsin usually, so we're looking at peptide mixtures really is our method. We use a chromatography to separate these mixtures in front of the mass spectrometer, so that's with this little micro LC, so it's small scale liquid chromatography. And as these peptides come off of a column, this big mixture, in real time, these things are flowing off a column, they're getting ionized, and these little colorful balls here are supposed to be those peptides. They get introduced into the mass spectrometer. The mass spectrometer is sort of taking a snapshot of measuring the masses of these ionized peptides, recording those, and then it's selecting and isolating a single peptide, putting it through the mass spectrometer in this fragmentation process. So it takes a full-length peptide, collides it with some gas, breaks it into smaller amino acid pieces, and then records the masses of those. And so what you get for every peptide is sort of this unique fingerprint of masses, these fragments that correspond to its sequence. And the mass spectrometer is just doing this over and over and over as this mixture of peptides gets fed through this instrument very, very quickly. So it's trying to sort of capture every peptide it detects, fragment it, and then move on to the next peptide in real time as these mixtures come streaming into the mass spectrometer. So there's sort of our core technology. So what we do is gather these tens to hundreds of thousands of these MS spectra, these fragmentation spectra, and now we try to annotate them. We try to put to them a sequence. We don't know the peptide that generated any given fragmentation spectrum. So to actually match a peptide sequence, we take it through this process of sequence database searching. So this is where software takes those fragmentation spectra, and now we use a database that contains known protein sequences, and we try to match those fragments to these sequences by generating sort of theoretical fragmentation patterns for all the known sequences in our database. And when things work out correctly, we get a peptide sequence that matches up to that fragmentation pattern. So this particular sequence of amino acids, if you break it apart in a predictable way, will create a fingerprint that matches this particular sequence. We can then say, okay, we found that peptide. We can then infer a protein identity based on going back to that database and saying if we found this peptide sequence that matches to this MS spectrum, which protein carries that. And usually if you're trying to do protein identification, you get more than one peptide in that mixture that you identify. So that helps you sort of infer that, okay, we found several peptides from this particular protein. It must have been present in that starting mixture. So we kind of build this data back up, and I try to identify what proteins are there. So sort of this peptide matching to a protein identification. Okay, great. So that generally works pretty well, has its limitations and pitfalls, but on the whole is a pretty mature technology. But if you're sort of paying attention to what I just talked about, you can start to imagine sort of, well, what are some of the limitations of this? One of those is that, as I said, the way that we identify the proteins in these mixtures is that we use a database of known sequences, right? So great, that's fine. We use something like UniProt, which is a pretty expansive database of known proteins, try to match things, and we call this sort of a reference database. But there's obviously a question about, well, what if my sample might have a protein in it that for some reason the sequence doesn't show up in this database? So it's a very good question. And so this is very much a contrived figure that I've made, but you may kind of come up with some real good reasons why your particular sample has this profile of proteins that don't totally match to what's in this database. Most of the things do, but there may be certain sequences within a sample, and that's what I'm trying to show here. It's a relatively small sliver that aren't in that database. So in the context of the talk, as I said, I called this the dark proteome, and Amy was saying, is this something to do with Star Wars or something like that? It's not quite sinister and evil like that. More so just, what about some of these things that are in this sample that we aren't capturing, and what are those, and why would we want to capture those potentially? And so one of the main reasons why there are certain sequences in a sample, any given sample, that may not be in that database is that we know that we're analyzing a cancer sample, a disease sample, whatever it is. There are mutations happening at the genome level. These could be SNPs, indels, you name it. We have certain gene models where we think we know which proteins get expressed from where within the genome. Those kind of go into these reference databases. But we know there's variations here which lead to variations at the RNA level. They may play a role in, say, splice isoforms and things. And in my relatively simplified sort of picture here of going all the way from DNA through RNA out to a translated protein, we know that these are carrying variants that are synthesized into a protein that may also then be an amino acid sequence that has been changed, whether it's a single amino acid change, it's a frame shift, whatever it is. And at least some of those proteins are going to survive and become an expressed functional protein, but we probably don't know about them in any given database that we're using. So these, you could make a case, might be some of the most interesting proteins and important proteins that maybe are underlying some sort of disease or health condition that you might be studying. So there's sort of the setup of, okay, great, we aren't capturing everything. How can we potentially be a little bit better at this? How can we capture some of these things? So this is sort of getting into this new sort of area that's been emerging for the last 7-8 years or so called proteogenomics. And really it's kind of a more, I'd say, intelligent or personalized way to do this whole proteomics type of experiment using mass spectrometry. And so here's the basic idea. We now live in this age where these high-throughput sequencing methods are out there. They're cost-effective. Most labs can do this relatively easily. And I'm going to focus at the RNA level. So you can take your sample, extract RNA, do an RNA-seq analysis, and you can get a pretty reliable sort of sequence across the whole transcriptome of all the RNA that's getting expressed, right? So what you can do then is say, great, we now have this RNA sequence within our particular sample that we're interested in. Let's computationally use that RNA as sort of a template to say, okay, here's the template that is potentially making all these proteins that are in our sample, and do a translation in silico of that RNA to create a protein database. You can do this in a kind of a variety of flavors. You can do it very unbiased, so you could do like a 3-frame translation and just say we don't even really know where and how this RNA is getting translated, where is it exactly starting, stopping, and just create all the possible proteins. You can also do things that are a little bit more targeted, like just look across that transcriptome and say look for regions when you compare them to a reference that are showing single nucleotide changes or are showing a frame shift or a splice isoform event, whatever it is, and then translate out all the potential proteins that come from those mutation events. So really you're kind of customizing this database now to contain proteins that you don't actually know if these are even being expressed, but you're saying these are possibilities based on this RNA template that are possible in our sample. So it's a little more specific, maybe a little more personalized database that goes with this sample that you're analyzing. So what do you do then? So now you take that same sample, and now we can do this sort of sensitive, comprehensive, high-throughput mass spectrometry analysis at the protein level. So isolate the same proteins, run them through the mass spectrometer, go through this whole matching process again. So same thing that I described. But kind of the key point here in yellow is that we're now using a customized database that is a little bit more intelligent. Maybe it captures more of the events that are occurring in terms of splicing isoforms or mutations, whatever they are. And this is just a good figure from a review a few years ago that kind of shows you what you can do with this information. So now we have peptide sequences. We match them. Lots and most of those sequences go to what we would say are reference proteins, things we already kind of knew about that are in the normal database. But other things are going to things that are now novel variants. So, and this just gives you a little snapshot of things where if we can map these then back to the genome, we get peptides that are matching to known gene coding sequences. We get peptides that map maybe to regions of the genome we haven't ever predicted. We're going to actually translate a protein in the end. Pseudogenes, also then we can start picking up mutations, single amino acid changes, and then also splice events. And that's what this is trying to lay out, is that in red would be the peptide sequences you've matched, and you may see a sequence that sort of spans and skips across the whole exon and shows you that there was some sort of novel splice event. So really giving you tools to kind of do this sort of more comprehensive identification of these proteins. Kind of jumping to the chase, I'll kind of get into how this is done, but this is just an example from a few years ago in a paper we published just trying to kind of proof of principle this sort of whole concept. And this shows just a certain genomic region of a couple of different proteins and their known coding areas within the genome. And when we map these things back, what we are seeing here, without trying to go into too much detail, is in red, all these sequences were novel frame shifts that were coming out of this coding region. In blue were sort of the known expected ones, which we also detected. But then as you do this, you start to see these other hits to things, and you map them back, showing, for whatever the reason in this particular case, where this was showing a lot of frame shifts of the proteins that were being expressed out of this region. So gives you at least a chance to start to capture some of these things as in terms of a profile. So let's go back to, okay, let's look at the practical side of this. Sounds great, cool, we got RNA-seq, we got proteomics, let's do this, right? So kind of for us, this was where we started and started looking at this and saying this would be really an interesting approach to take, but immediately you run into this problem of, and all I'm trying to show here is get across that there's a lot of steps and complexity to this, because now we got RNA-seq data, we got proteomics data, we need to generate a database from the RNA-seq data, and then we need to match proteins, and we need some ways to filter through that data. So it becomes a pretty complicated just bioinformatics challenge, because there's a lot of different pieces of software that somehow you got to master and bring together to actually do this. So that was sort of a starting point. Is there a way that we could actually start to do this to make this a little bit more of a routine approach? So I put this in here because this is one possible approach. Maybe you hire a lot of people or some monkeys here, and they each take a little piece of this and learn one of the programs or something and pass that off to the next person. I was going to make a joke about maybe graduate students versus monkeys or something, but I don't really want to go down that route. I think grad students feel like monkeys in front of a computer sometimes. But so, okay, there's one brute force possibility of just let's figure out all these pieces of software and figure out some way to deal with it. And so we were kind of thinking about this. It kind of went with all kinds of other things where a lot of our data from mass spec was just expanding. We had more and more needs of it. We had been working with the Supercomputing Institute already in terms of transferring data, and they were helping us analyze it and putting software in place. And we were trying to then think about, well, should we, we got all these interesting applications. Do we need to sort of develop our own platform here to bring all this software together? What can we do? So we were kind of pondering that. And at that time, Anne-Francoise Lamblin was over at MSI, who some of you may remember. It's not here at the university anymore, but had started to put in place this platform called Galaxy, which is this genomic data analysis platform. And she kind of at that point sort of said, well, why don't you guys think about, extending and enhancing Galaxy to do proteomics and other things? That was pretty much what I looked like at that point and said, what are you talking about? I have no idea what this is. So kind of got a little bit more in tune with it. We rolled the dice, and that was when we threw in a grant, which NSF decided, hey, that's actually a pretty good idea, and it sort of has taken us down a whole new path. So without getting too far into what's Galaxy, I think some of you are probably familiar with it. It's the platform. If you take things through the UMGC here, your data gets shuffled over to the Supercomputing Institute, and they use Galaxy to help assemble and analyze the sequencing data you do. And so what it is this software, open, community-based, developed over 10 years ago now by this team that was at Penn State and Emory, at that point. And so it's this meant to be this interface and this platform that was designed kind of in the same idea of, in the genomic data analysis area, there's all this software out there. Can we make a platform where we can kind of capture all this software in one environment, make it so that it's automated and usable, and that bench scientists can actually learn it and use it and bring all this software together? So it actually made a lot of sense for us, and kind of in a nutshell, and I'll show you a little of this, is kind of this, I think, paragraph kind of sums up what Galaxy is trying to do. And really for us, a lot of it is this idea of disparate software programs, where we have lots of software, and it gives us an environment to put this software in, integrate it into these workflows. We can do things like sharing and reproducing it and all kinds of different things. I put this in here because at the outset of this, and we got into it, and still are this way, there was already a lot of software out there, proteomic software, all these and really good labs making this, and we were jumping into this. So all along we've said, we're not trying to reinvent the wheel. We're not trying to make new software programs that somebody also already has. But really what we're trying to do is bring those into a single environment. And I don't know, this was my analogy of taking the wheels, not reinventing them, but maybe we're putting them together to make something that's a little bit more bigger and more valuable. What we do isn't probably as sort of pretty as that Ferrari is there, but probably doesn't cost as much as that Ferrari either. But so that's really kind of the overarching idea. So as I said, some of you may be familiar with Galaxy, but it is this web-based portal. So it sits, say, on a server, where this software gets integrated into the main Galaxy instance. You as a user then are registered into that instance, and you can open up a web browser, go to the right address, and go find that Galaxy instance, open it up. Gives you this sort of look, such as this tool menu, so all the software that's been implemented and tested and ready to go is there. You can now take those tools and integrate them into workflows, and hit go, and off it goes, and it runs, and it shows you sort of an active history of here's what your analysis is doing. So just a real brief look at what Galaxy is. And really what I think is probably as strong as anything is, as I said, one of the first things is that it gives you this environment to pull in lots of useful software, integrate those tools together to make a pretty sophisticated workflow. And then one of the real nice things with Galaxy, and really how they designed this from the start, is that it gives you the ability to save that whole workflow. So you may have worked a long time on parameters for all this software and optimizing it, and rather than just sort of have this, written down in some methods part of your paper, what you can actually do is save that workflow in Galaxy, and then you can share that with other people. So you can send a link to other users, and they can actually go and import that workflow. It's completely how you made it to be. They don't have to know every parameter, and they can pretty much bring that in, learn a little bit about it. If they have the right data to put into it, they hit go, and off it goes, and it runs. Never fails, always works, right? Once in a while, something goes wrong, but as I've learned, software has got its own issues sometimes. But anyways, that's the concept. So we've been sort of developing then, and I would say extending what Galaxy is and trying to take it into new directions. And I'm talking today about proteogenomics. I just put this in here that we have a couple other outgrowths of it, or application areas that we think Galaxy is very well suited for. Metaproteomics, which is sort of a complement to metagenomics, so looking at microbial communities within samples, but actually looking at proteins that are expressed. So we've been doing a lot of work in trying to kind of have some of the similar challenges that proteogenomics does, is bring tools and software to do that. So kind of an exciting area, and pushing towards maybe using some of these tools more in a disease-relevant setting with microbiome contributions to a host, where you can now analyze proteins from the bacterial component. So we're trying to kind of put in place some tools that help there. And a little less mature, but still something we've been working on, is this metabolomics area. So with Adrian Hegeman, who's here over in St. Paul, actually, and Art's a postdoc, Galaxy also gives you a really nice platform to bring together all the tools that metabolomics needs, because that's still such a sort of young and emerging area. So we're doing some work on putting some of those workflows in as well. But for today, we're focused on proteogenomics. So what I'm going to do is just kind of take you through, as I said, kind of through the pipeline of what proteogenomics does. I'm not going to get into the weeds of each piece here, but give you at least sort of a high-level view of why this actually is taking the length of time that it is. So you might think, well, great, you got Galaxy, you know the software, you know what you want to do, let's just throw some software in Galaxy and everything would be good to go. As I've learned, and we've learned, this still takes some time in terms of development, and it's been ongoing. But I'm going to kind of take you through the main sort of modules that make up, say, this proteogenomic workflow. And so this first slide, and this is actually a pretty sort of simplified slide of what's actually going on in this workflow, but it kind of shows you the main pieces of it. So this is the first step, is to take RNA-seq data and generate this protein database, which seems like it might be somewhat straightforward, but as I've certainly learned, it has a lot of components to really do this well, and then have it made so that you can do things with it downstream. So what we have been working hard at, and are really kind of close to, I think, having sort of a optimized, validated workflow for this, is these sort of two parallel workflows where we take in RNA-seq. We always are utilizing a reference genome, as well as this is a file that gives you information on gene structure for the genome that we're analyzing, this GTF file. But we're pulling in that RNA-seq data, which is the raw data that we would have generated experimentally. And we're using these tools to align that and compare it to a reference genome to know where all those reads go, and kind of standard RNA-seq analysis. And then those go into some other RNA-seq tools. And in this workflow, this is a workflow where we are actually trying to pull out of that RNA-seq single amino acid variants, that's SAVs, short indels that might be happening. So we have this sort of specific workflow to trying to find those sequences from the RNA and then translate them into proteins and create a protein sequence database that now captures those types of potential variants. This is the other workflow, and I'm calling it other variants because it's trying to capture a wide variety of things. Things like splicing events, frame shifts, places where potentially sequences of peptides that are coming from unexpected regions of the genome that are getting expressed. And so this is a little bit newer, but this is what JJ has been working hard at, and he uses this thing called string tie to assemble and try to find some of these different types of variants and annotate them, and then ultimately translate those variants to proteins, and again, put them into this protein database. We're also capturing with all of this genomic coordinates of these proteins, because later we want to map these things back to where on the genome they came from. So a number of layers of sort of detail that this overall workflow is trying to capture. And this is that Praveen, who's a graduate student, actually made this, and I think it's a pretty good attempt at trying to just sort of capture all of the variant types that we're trying to potentially see in these. I'm not going to try to go through all of these, but it kind of gives you an idea of not only are we going to find known proteins, and if this is just an example, if in a known coding region, we're going to find all kinds of sequences that map back to known coding regions exactly like we'd expect them, right in the right frame. That's the majority of the things. But then what we're trying to do in these databases is capture all of these sort of other possible events that might be happening from splice isoforms that cross different exons and are evidence of some sort of novel splicing. All the way down here, this is a single amino acid change that's in the star, so normal sequences, but now have been shifted by a single amino acid, as well as other things where you're getting translation out of unexpected regions and things that you maybe didn't think were actually leading to a protein product. So that's where we're kind of starting. The second module then is actually identifying, matching the mass spectrometry data to these sequences. So here it's relatively routine, but even here there's sort of a lot of questions. This is a very new area as to how you best do this. Do you just take a massive database of all these sequences and throw all the data at it? Or some of the things that we're trying and have been sort of postulated as maybe better ways to do this is something that, again, Praveen Kumar has been working on, where maybe you divide these databases, and rather than one big one, you have more variant-specific databases of certain types of variants in each different database, and then you do sort of sequential matching of your data where you see how many things match to single amino acid changes. Whatever is left over now you throw at a different database that has other transcript variants in it. Gives us maybe a better chance to do things like control for false positives and things like that. So fair amount of complexity there, but again, here we're using a lot of standard things that are out there. We use something called Search GUI, which is a program that does this matching that's been out there. It's an open source program, uses a number of different algorithms to do that, so to try to give us more confidence in the matches, and then brings it all together in this tool called Peptide Shaker. So again, this idea, we're not trying to reinvent those wheels at all. We're bringing those into this, trying to use them sort of in a new context. Okay, so the third module. So now you can do this and you can get identifications of peptides right out. And so now you have these matches to sequences that they might be pretty confident and all of that. Turns out that there's a whole nother level of caution you have to take here, because then there's this question of, we need to make sure if we think we're seeing a variant peptide sequence, is that really a variant? So we actually take it through this step where we have implemented the BLAST-P tool within Galaxy to automate this so that you can take, say, a list of things we think are variant sequences based on how we built these databases, but just as an extra caution step, take them through a BLAST-P search. And in this case, you might be familiar with BLAST-P. It's looking for sequences within a big, large database of known proteins that match the one we've input to it. What we're actually doing here is we're looking for any of these variants that when you do the BLAST-P sort of survive this because they don't match anything within that database. So another sort of level of caution of filtering out only things that are actually, as far as we can tell, novel peptide sequences that nobody else has actually seen before, so that we can actually have some confidence in those matches. And then last, which is something we're actually working really hard on, is how do you get this data and use it? So how do you visualize it? What do you do with it from there versus just, a tabular Excel file, which is fine. Trying to give a user, especially somebody who's a bench scientist, the way to look at this and interpret it and do some things with it. So we're working on something we call the Multi-Omics Visualization platform, and I only give sort of a brief snapshot here, but once you do get the results, this is what's called a plug-in to the Galaxy. It opens up this visualization portal, so to speak, and it gives you the ability to do things like, here's our list of peptides we identified. Maybe these are just peptides that are variants that we've filtered out. You can look at the actual fragmentation patterns and make sure those things look good and are of high quality. You can then look at the overall protein sequence where this peptide came from and which other peptides were identified, as well as we're really trying to make this a sort of a genome mapping tool too. So you see the sequence, but then it also gives you some information on where in the genome do these sequences map back to, what's the nature of the variant that you've seen. Is it a single amino acid change or whatever it is? And then we also are utilizing IGV, so very well-known and well-used genome browser tool, where you can actually click right out of this visualization tool, our protein visualization, and open up IGV and now look at that sequence mapped back to the genome in higher detail of what was the RNA sequence that this came from, where exactly in the genome does this protein come from, and all those types of questions. So trying to put in the hands of the user some kind of valuable tools that they can really start to interpret and visualize their data with. So, and I just put this in here as another piece of this from the start, and really I think an important key to actually getting a funding for this project that we've been doing is we've really had an emphasis on dissemination of this and training, so that this isn't going to be software that runs on a computer in our lab and that's about it. Nobody else knows what to do with it. But we've been trying to do things like we've set up in the cloud, as everything is happening in the cloud. And this is part of a paper that we published. It's sort of an accessible and public portal. So this is an instant. It's actually running on this thing called Jetstream. That's an NSF-supported computing infrastructure. We're working with people at Indiana University. So sitting on some high-powered server of theirs is this Galaxy instance that has software that we've validated and put into it that people can come into and register to use and start to actually try to use these along with training material and things like that. So really trying to kind of push this out to get more people adopting the software. All right, so I only have a few more slides, and this was kind of a few things that are sort of more ongoing or sort of pushing into the future. You may ask very logically, great, you're identifying variants. Well, what about quantifying these things? So anybody doing a biological experiment always wants to compare conditions or compare disease versus control, whatever it is. So we're in process of putting that piece into this, and one of the main pieces of this is what we call label-free quantification. We're actually working with a couple of collaborator groups, one in Belgium and one at UW-Madison, who have developed some software to do this and trying to get it implemented into our Galaxy workflows. So label-free basically is very much amenable to sort of like this proteogenomics approach where we're seeing variant peptides, and we may want to go back in and say, okay, across a number of samples for that variant, what was the profile? Does it change across a disease state or a time course or whatever it is? So label-free quantification basically goes back into the data. You have a peptide sequence, you know the mass, and it extracts that particular peptide from the data that the mass spectrometer recorded. So this is liquid chromatography mass spectrometry data. And it basically reconstructs the signal for any given peptide across the liquid chromatographic peak. So it kind of looks like a UV trace if you were using an HPLC, but we're using mass instead. And it lines up then that peptide across these different samples and how much of that peptide was there, and starts to give you the ability to get quantitative information. Has all its own trickiness in terms of how to do this well and confidently, but that is definitely one place that we're working on some things. We're also working on some things that are trying to get a little bit more statistical confidence to the idea that when we do identify what we think is a variant peptide sequence, you really want to have tools in place that ensure that you're really calling these things confidently. So there's a lot of possible pitfalls in this whole database search matching process that can give you false positives. So without going into details, we're working on some sort of more sophisticated algorithms where we can look in green at those things that are the true positives and make some cutoffs on this is the type of data that is most confident that you really should follow up on, whereas these hits, they're a little bit shaky. You want to be careful. Just kind of giving, again, somebody who's using this hopefully an end result that they can interpret and use a little bit more. And then sort of one last thing that we're working on the informatics front is, again, sort of an interpretation tool. And how do you, once you get this information, how do you take this information as a user, as a biologist who I just want to get a result that I can do something with, right? How do you interpret this? So this is actually through the NCI program that we're funded through. We've gotten to know this group that has made this cravat tool, and some of you maybe have actually even used this. It's actually really a lovely piece of software. it is really meant to take RNA-seq data and take variants you've identified, say, in RNA-seq data, put it through this program, and then it goes and queries things like Cosmic and various databases and gives you back some things like what is known about the variants you've identified. Do they have pathologies that are associated with them? And so we kind of saw this, and they've been developing this in the last few years and said this is actually perfect for a proteogenomics workflow because we're not necessarily identifying RNA sequences, but we're identifying proteins, which similarly we want to ask the same questions. What gene do these come from? What's known about their pathology? And this is just a very, I know, small snapshot, but Ray, who is in the group, has started working on sort of an endpoint for our workflows where we can pop open some of these results in this tool, and it starts to give us things like, this is sort of what the gene structure and where on the protein where, say, the amino acid sequence change was, and then it goes to actually like the structure of the protein and says, well, this is where in the known structure of this protein your mutation happened. So really a nice tool that we're trying to kind of implement into our workflows and learning a lot about still, but I think is going to be kind of interesting to use as we go forward. And then I just have a couple last slides because I'm not going to get into a lot of depth on this, but there's also then this question, I think anybody, if you've done mass spectrometry-based proteomics, you might be thinking this. Okay, I do mass spectrometry. We look at these peptides, right, and we get sort of a snapshot of the protein, but as I said, in red here might be, you know, if you're doing okay, you might get 25% sequence coverage of your particular protein you're analyzing, right? And that's fine if all you're trying to do is make a case that that protein is present. But what if we want to really do this sort of in-depth profiling of protein variants? We're missing, and that's what I put in green, we're missing a lot of peptides that don't fly well or whatever the issue is. We just don't see complete coverage of a protein, right? So certainly is a limitation if you're trying to make a case that we're finding protein variants. We're going to be kind of only seeing maybe half of the picture or something like that. So I don't want to get too deep into this, but what we've been working on, and one way to sort of improve that, is changing the way we do this chromatographic separation. So usually we use reverse phase chromatography. It binds peptides based on their hydrophobicity, and they come off as you increase this organic mobile phase over time. Works great. It's sort of the standard way. A number of years ago, we started playing around with a different chromatographic phase called ERLIC, electrostatic repulsion hydrophilic interaction chromatography. It's a big mouthful, but it kind of flips reverse phase on its side, and it actually is a chromatography method that more hydrophilic peptides actually hold on to this longer, and it actually separates peptides based more or less on isoelectric points. So basic peptides are retained less and come off earlier, and then the more acidic peptides come off later, and you actually do, rather than an organic increasing mobile phase of organic, you do aqueous. So a very sort of orthogonal methodology. So truthfully, we started trying to play with this just out of curiosity and saw that what's interesting about it is if you take the same sample of tryptic peptides, you run it through a reverse phase into the mass spectrometer, and then you run the same thing through ERLIC into the mass spectrometer, do the same tandem mass spectrometry fragmentation process, is that these are really complementary. And this is just a snapshot of a little data that we've been trying using ERLIC versus a reverse phase, the standard approach, and then taking it through proteogenomics and saying, what do we identify? And what we actually see is that if you do ERLIC in combination with reverse phase, you get this very complementary profile. So basically what we're doing is increasing that coverage of the protein sequence by doing this other phase of separation and introduction into the mass spectrometer in parallel then to the normal way. And where we're trying to go here is potentially maybe use more than one enzyme. So you can also take, say, trypsin and a different enzyme like elastase. These cut the protein up at different parts. Run these also as a mixture, and when you do the matching, then you kind of get more kind of like, looks like sequence reads that you can start to lay on top of each other to get a more complete sequence of the protein. So still in process, but it is one thing that's at least in the thought process here of, okay, you can get all these wonderful bioinformatics tools. How can we improve coverage of the actual proteome? And sort of for my last dream slide, you know, I think there's a possibility as all these things come together that we do genome sequencing now. You get this sort of genotype at the genome level, which is terrific, obviously has tons of value. But could we get to a point where not only at the genome level, but out here with these translated proteins, we can get a pretty comprehensive sequence of those proteins, including these variants that are specific to any given sample, so that you get maybe a proteotype that is associated with disease or cancer. As a protein biochemist, I'm all about what's going on with the proteins. I think this could have tremendous value potentially. So still need the technology to get there to do that in a real sort of practical way, but I think there's some potential there. And my last slide, just want to circle back around. Again, this is a lot of work by a lot of different people, but I highlighted on here, this is sort of a list of, I would call the biomedical collaborators, many of which who have cancer-related types of projects going on. But the work that we have been doing, really it's driven by these projects because we need real data, we need real projects to help understand what are the tools and what's the nature of the tools we need to make these tools practical and useful. So as I said at the outset, I was trying to hopefully give you a little view of what we're doing, and we are always very much looking for people who, especially if you're already planning an RNA-seq type of experiment in your sample and have the potential to also combine that with proteomics, we have the tools here that are pretty much already in place to start working with those types of samples. And a number of these collaborations, I'll highlight just one, Jiang-Sik Yang, they've developed a way to tease out of RNA-seq data these very novel transcripts that now we're trying to help them develop ways to identify the proteins that might be expressed from those. So it's a really nice potential application to, if you have a sort of indication that you have some sort of novel gene expression, novel sequence, we can help try to actually give some confirmation at the protein level that these things are turning into a functional protein. So I'll leave it there. Happy to take some questions.
We'll start in this room, and then we might have some questions in our other locations.
Thank you, Tim. That was a great talk. I have a question about the last slide that you had put up. So some of us are doing rather extensive. Yeah, so some of us are doing DNA and RNA level analysis of tumor tissue, and in those cases, we're sometimes limited by the amount of material we can get. Sometimes it's a biopsy, sometimes it's circulating tumor cells. What scale is the proteomics at, and what's needed to fill that gap when you have limiting amounts of tissue available?
It's a really good question. It's certainly still a limitation at the proteomics side. So it's gotten better because the instruments are more sensitive, but we're still in the realm where you need to reliably be able to get maybe in the tens of micrograms or so of total protein out of a sample to really, at the way we're approaching things now, to really get pretty in-depth data. It could go down less than that. I mean, on the instrument itself, if you have a microgram or less to load onto the instrument, you can get an expansive, thousands of proteins. Problem is, if you need to do some preparation up front of that to get the thing ready for the mass spec, and you're only starting with that much, then you lose stuff along the way, and it gets more limited. So there are a number of groups, though, that are working on pretty advanced and still very research level methods to do like single cell proteomics where you have very small populations, trying to kind of work out micro scale preparation of those to introduce them to the mass spectrometer. So I would hope those keep to advance and come to a point where the masses like us could maybe start to adopt those a little more too. So, but it's not for every sample at this point. If it's really limited, it can be definitely difficult.
Other questions or comments?
Hi, I was just wondering, since there's a lot of RNA-seq data that other groups have already published, have you or other groups already taken it and put it into that big proteomic database so that we could just search using our samples?
It's a really good question. Yes, we haven't done it. There are some groups that have been actively, and there's databases out there that they call them like the cancer or something database and this and that, where they've tried to just collect from RNA-seq data or other evidence, whatever it is, protein databases that now contain sort of known and validated sequence variants or something. So there's definitely a move on to do that. You could see it more large scale potentially, where you take the TCGA RNA-seq data and turn it into a protein database. There's room to do that. Nobody has started to do that on a large scale, but there are smaller efforts like that out there.
So many of us are interested in post-translational modifications.
Mm-hmm.
And so is there a specific place to, in the pipeline to do that? Is that already part of the development? And
yes. Yeah.
What are the ones that you can readily identify
Sure.
at this
Very
point?
good question. So post-translational modifications are definitely part of the story. So we can do what is kind of the standard way to do things, where if you think phosphorylation events are happening or whatever it is of pretty well-known modifications, when you do that database searching, you add those in as parameters, and it looks for mass shifts that might correspond to those. Usually, if you want to really do that in depth, you need to do sort of an enrichment of like the phosphopeptides first and then analyze those. And then you see lots of phosphorylated things versus just a big mixture. You'll see some hits, but not that much. It's still a challenge to get really good in-depth PTM profiling. There are some of the groups we're working with, and we haven't gotten to really implementing them, are doing these things now where you can do that matching process and you have this sort of wide mass window where it allows for, say, unexpected mass shifts on any given peptide. And then you still get a very confident hit to a sequence, but it also tells you that this sequence had this shift on some amino acid of some mass. It may correspond to something that's known, maybe something that's completely new. So there are some more sophisticated ways to pick those up, but it's a really important part of this. And we're also trying to do that because one thing that can happen is when you're looking for like a single amino acid variant, you could also have a PTM on a normal sequence that sort of mimics the same mass shift as if you change one of the amino acids. So some complexities there where definitely is in our interest to also try to pick up PTMs so we're not missing those or miscalling other things. So it's an important piece.
So I was curious, you mentioned you use Ehrlich in the standard reverse phase, and they're pretty complementary. And so does that sort of take the place of doing sort of an orthogonal fractionation of your peptides prior to the reverse phase?
Yeah.
Or do you still have to do that kind of
Yeah,
stuff?
good question. It doesn't necessarily take place of that because it's not going to, you do that like offline fractionation or something to give you more sensitivity and depth for lower abundance proteins. So if you want to get those, you still probably have to fractionate it somehow prior to running them on reverse phase or Ehrlich to really get deeper into your sample. What is occurring, though, is the instrumentation is getting more sensitive to the point that all that upfront fractionation is starting to get a little less needed because the instruments are so sensitive. And now you can maybe draw out the length of the chromatography time, and it starts to give you actually a chance to really get at lower abundance things without having to do all that upfront fractionating and all that. So we're pushing towards that and getting more, hopefully, to more high throughput proteomics where you can do lots of samples and you don't have to go through all of that drama up front of it to make it work. But not totally there in routine use yet, but that's where I hope we're going.
Other questions or comments? Nick, anything from anyone else? Okay. All right. Well, thank you very much.
Thank you.